Chromosomal microarray and whole exome sequencing are cornerstones of modern genomic diagnostics. Each offers unique insights. Chromosomal microarray identifies larger structural changes like copy number variations, C and Vs. While exome sequencing investigates the exome, the protein coding region, where most pathogenic mutations reside. Chromosomal microarray is a frontline tool for neurodevelopmental disorders, intellectual disability, and congenital anomalies. It scans chromosomal imbalances missed by karyotyping, offering around 15 to 20% of diagnostic yield in such cases. Its speed and cost effectiveness make it a go-to for initial evaluations. Whole exome sequencing, by contrast, dives into the exotic 1 to 2% of the genome that harbors around 85% of non-disease causing variants. Whole exome sequencing is particularly useful in cases where chromosomal microarrays results are negative, especially when a monogenetic disorder is suspected. It excels in detecting single nucleotide variants, indels, and gene-disrupting mutations. But may miss structural rearrangement or low-level mosaicism. Chromosomal microarray is strong for copy number variations, limited for point mutation. While whole exome sequencing is strong for sequence variant, complex interpretation. Whole exome sequencing is best used after chromosomal microarray in unresolved syndromic cases. The combined use improves yield, informing diagnosis, genetic counseling, and treatment. Whole exome sequencing presents challenges around incidental findings and variant uncertainty, requiring robust genetic counseling. targeted may still identify risk alleles with familial or reproductive implications. Chromosomal microarray and whole exome sequencing are not rivals. They are complementary technologies. Together, they represent a genomic toolkit that supports precision diagnostics, enhances personalized medicine, and shapes the future of ethically patient-centered care.